Hello and welcome to my presentation on human immunity. Today I'll be talking about non-specific defences against diseases, specific immunity, and the biology behind vaccines. First I'll be talking about infectious diseases. These are caused by pathogens, which are any organism that causes diseases. Pathogens include bacteria, viruses, fungi, protozoa, prion, and parasites. They are either acellular, non-living, or cellular living. So first we have bacteria. This is cellular, single-celled organisms. These cause illnesses such as cellulitis, strep throat, and syphilis. Viruses are acellular. They're an infectious particle that reproduce by commandeering a host cell and using its machinery to make viruses. These range widely from HIV to cold. We have fungi, that is cellular. They're eukaryotic organisms that include microorganisms such as yeast, molds, and mushrooms. These cause many skin diseases such as athlete's foot, but also lung and nervous system infections. Prions are acellular, abnormal pathogenic agents that are transmissible and are able to induce abnormal folding of specific normal cellular protons called prion protons, most abundantly in the brain. These cause diseases such as CJD. Protozoa are cellular. They are unicellular, eukaryotic, heterotrophic organisms. They are either free-living or parasites. These cause infections such as malaria. Parasites are cellular and they're highly specialised organisms that derive food from the host at the host's expense, such as tapeworm. There are many methods of transmission, such as direct contact, which is person to person, animal to person, mother to unborn child, and there's more examples of how those occur. Insect bites, indirect contact, such as germs on an inanimate object, or food or water contamination. First, lines of defence. The body has 3 lines of defence. The first 2 lines are non-specific responses and the third is a specific response. Surface barriers that prevent pathogens from entering the body are the first lines of defence. These include skin and mucous membranes. Here is a diagram of the skin. The skin is the largest organ protecting the body from pathogens. It prevents most pathogens or infectious agents such as environmental toxins. As long as the skin remains unbroken, it's very effective. So the integumentary system includes the epidermis, dermis, and subcutaneous tissue. The epidermis acts as a barrier by preventing or at least obstructing penetration of microbial organisms, chemical irritants, or toxins. It also absorbs or blocks ionized solar or radiation, and it inhibits water loss. Mucous membranes are also found in circles of eyes, nose, ears, mouth, throat, gastrointestinal tract, respiratory tract, urogenital tract, and vaginal tract. These are commonly coated with secretions that fight microorganisms, therefore preventing pathogens from entering the body without inhibiting the body's healthy status. Mucosal layers can be compromised, inhibiting them from acting as a layer of defense. There are other chemical and physical barriers acting in certain areas of the body, such as the gastrointestinal system. So the digestive tract contains multiple barriers: stomach acid, pancreatic enzyme, bile, and intestinal secretions. The stomach pH is too low for most bacteria to survive. Small intestine is pH is much higher, but it's still able to kill most pathogens other than cholera and typhoid. And then contractions and the shedding of cells from the lining help remove harmful and dead microorganisms. That's a natural, normal process. Nasal cavity. The walls of the nose and airways are coated with mucus. Microorganisms become trapped in the mucus and that is removed by coughing or blowing the nose. Rhythmic movement of the cilia helps remove trapped particles in the lungs. In the urinary tract, the bladder is protected by the urethra. In males over 6 months old, the urethra is so long that bacteria rarely makes it to the top due to frequent passing of urine. Unfortunately, women do not have that luxury and the urethra is a lot more short, meaning that UTIs are much more common, especially in pregnancy where progesterone loosens or relaxes various tissues, making it much more common. The female genital tract, the vagina contains lactobacilli. This inhibits the binding of other bacteria to epithelial cells and produce lactic acid that kills or inhibits the growth of many other bacteria. A healthy pH of 4.5 prevents harmful bacteria growing while maintaining healthy protective bacteria. The second lines of defense includes inflammation, fever, phagocytes, complement proteins, and cytokines. Inflammation is a localized defense mechanism. Symptoms include red colorization, swelling, pain in the affected area, and also increased body heat in the affected area, which causes an antibiotic effect. As you can see, here's a diagram of injury inflammation. So damaged mast cells will release chemical signals of histamine. Histamine causes vasodilation, which is dilation of the capillary and blood vessels in the local area. This results in increased blood flow, and antimicrobial proteins such as chemokines, fluid and platelets exit through the capillary. Clotting then closes the wound and stops bacteria from spreading. Chemokine concentration will be highest at the point of injury, providing a map for phagocytic cells attracting more to the injury. Neutrophils and macrophages phagocytose, which is engulf and destroy pathogens and debris from the site, creating pus, swelling, which causes the pain. Tissue then heals and histamine levels will reduce to normal. So phagocytes are neutrophils and macrophages. They perform phagocytosis, the engulfing and removal of dead cells or invasive microorganisms. Once engulfed, the phagosome will fuse with the lysosomes creating a phagolysome. Lysosomes contain digestive enzymes which will disintegrate the contents of the phagosome. So here we have the two. These are under the microscope as well. So we have phagocytes, which are macrophages and neutrophils. So macrophages are formed through differentiation of monocytes. They're very versatile as they can modify themselves to combat specific infections. They detect, engulf and destroy pathogens, and they're long-lived cells. Neutrophils are much smaller than macrophages. They're short-lived cells. They're the most common white blood cell, and they can move through blood vessels by chemotaxis. They're usually the first at the site of infection. When the body experiences infections or damaged tissue, monocytes leave the bloodstream and enter the affected organ. So here we have cytokines. Cytokines are a type of complement protein. So the presence of cytokines attracts more cytokines. Here are some examples of cytokines. So complement proteins are a group of over 30 small proteins. Most of them travel when active. They're activated by triggers such as the presence of a pathogen. Once activated, they interact with each other, forming a cascade of reactions. So now we'll talk about the third line of defense. Antigens are parts of pathogens that cause an immune response in the body. They're needed to infect the body in the first place as they help viruses enter the cell. So these can be any type of molecule, such as proteins, carbohydrates, nucleic acid, and fat. So we'll just quickly talk about acquired immunity. Acquired immunity is not present at birth. It's a learned response from the body's immune system. The three main points is learn, adapt, and remember. So if someone's exposed to a natural environment, the immune system will encounter antigens. So artificially acquired immunity can be active or passive. Active is when the body is intentionally exposed to a foreign antigen in a vaccine. Passively is when the body is introduced to preformed antibodies in order to generate protective immune activity. We'll talk about that a bit later. Both require human intervention. So here are 5 of the immune cells. These are leukocytes. They are white blood cells. All 5 are produced in the bone marrow. We have monocytes, lymphocytes, neutrophils, eosinophils, and basophils. Lymphocytes are T cells and B cells. T cells mature in the thymus gland. There are 3 types of T cells: cytotoxic T cells, suppressor T cells, and helper T cells. Helper T cells make up 75% of T cells. Suppressor T cells suppress cytotoxic and helper T cells, reducing the ability of having they regulate the immune system and protect against an autoimmune attack. T cells cannot detect foreign substances themselves. They require help from antigen presenting cells, APCs. APCs can communicate with T cells due to a special type of molecule on their surface. APCs break down foreign substances, presenting it to the T cell so that they can mount a response. A response is then activated, and lots of different types of T cells are released into the bloodstream to destroy the foreign substance. The key roles of T cells is signaling for growth and activation of B cells, signaling for growth in other cells, stimulation of cytotoxic T cells during viral infections, and activation of cells that can eat foreign substances. So B cells make up for 10 to 15% of all lymphocytes. Maturation takes place in the bone marrow. B cells release a huge number of antibodies when they are activated by helper cells. They have 2 major roles: antibody production and presenting antigen to T cells and providing signals for T cell activation. There are plasma B cells, and memory B cells. Plasma B cells are short-lived and they produce large amounts of antibodies against antigens directly into the circulatory system. Immunity is maintained as long as the plasma cell continues producing antibodies. Memory B cells can be formed after stimulation. When they are not actively engaging, they remain in the lymph nodes. If you are infected by the same pathogens, memory B cells rapidly differentiate into plasma B cells quickly to produce antibodies against the pathogen. Immunoglobulins are sometimes referred to as antibodies. Most immunoglobulins consist of 2 light and 2 heavy chains. They have an antigen binding site and an immune cell binding site, as shown in the diagram. Some of their roles include targeting infections, neutralizing toxins, activating mast cells, removing antigens, complement activation, recruitment of immune cells, and stimulation of cells that can eat foreign substances. There are different types of immunoglobulins as shown in the picture. First is IgG. This is the most abundant. It's a major activator of the complement pathway and it stimulates a direct attack on anything recognised as foreign. IgG is the only Ig that can be transferred to mother to unborn child across the placenta. IgA is the second most abundant. It has an important role in the cellular defence of soft mucosal surfaces. Lack of IgA can cause major infections in mucosal surfaces. IgD is uncommon and its true immune system function remains unknown. IgE is the largest Ig. It's present in low levels in a healthy individual. Levels increase significantly in response to an allergic reaction or parasitic infection. IgM is the first Ig synthesized in immune response and it's made up of 5 normal Igs. So, self and non-self. The immune system recognises non-self and self through antigens. So for example, if an antigen on a red blood cell is encountered, it's recognised as self. However, as soon as one is recognised as non-self, an immune system response will be activated. Antigens very similar to self will have a much weaker immune system response than those very different. So the major histocompatibility complexes 1 and 2 allow the system to distinguish between non-self and self, along with human leukocyte antigen gene complex. MHC1 molecules are expressed on all nucleated cells, and it's an early warning sign for the immune system. It allows foreign proteins to be displayed on the cell surface for T cells to recognise. MHC2 is only expressed on some cells compared to all nucleated cells. It's extracellular presentation mechanism and it allows antigen fragments to be picked up and displayed on the surface for T cells. You can develop hypersensitivity. This is abnormal reactions due to repeated exposure to an antigen. Autoimmune diseases are an example of hypersensitivity diseases and these result in chronic problems. So here are some of the hypersensitivity reactions. You can have immediate type 1 hypersensitivity, antibody-mediated type 2 hypersensitivity, immune complex-mediated type 3 hypersensitivity, and cell-mediated type 4 hypersensitivity, as shown here, and how they are produced. So the immune system produces 100 million different types of cells. It's constantly destroying self-reactive cells. If this fails, B cells or T cells that react to self-antigens may be produced, which is the basis of an autoimmune disease. So next we'll just quickly talk about the rejection of transplanted organs. The immune system is very effective against battling against foreign bodies. The transplantation is the process of cells, tissues or organs being transplanted from one site to another. You can prevent rejections by antigen matching, tissue typing or monoclonal antibodies. There are different types of reactions, such as the hyperacute reaction. This happens a few minutes after. If the antigens are completely unmatched, the tissue must be removed and it can result in death. Acute reaction happens the first week to 3 months after. Every recipient will have some kind of acute reaction. Chronic rejection happens over many years. Its constant immune response damages the transplant. So next we'll talk about 2 different types of autoimmune diseases. First is rheumatoid arthritis. Rheumatoid arthritis is an autoimmune disease usually affecting hands, knees, feet and wrists. It can affect joints, cartilage and nearby bone. Symptoms are pain, swelling and stiffness in joint, tiredness and weight loss. Diagnosis includes a physical examination, blood tests such as ESR, CRP to assess levels of inflammation, a full blood count to rule out other causes, and rheumatoid factors, which can be tested for the levels. These are proteins that are produced when the immune system attacks health tissues. You can also get anti-CCP tests, which are antibodies produced by the immune system. However, you can test positive for these and not have arthritis. Treatment includes disease-modifying anti-rheumatic drugs, painkillers, physiotherapy, occupational therapy, and nutritional supplements and dietary changes. Lupus is a long-term autoimmune disease causing joint pain, skin rashes and tiredness. Symptoms can range from mild to severe. Mild is joint and skin problems with tiredness. Moderate is inflammation of parts of the skin and body including heart, lungs and kidney. Severe is damage to the heart, lungs, kidneys or brain from inflammation. Blood tests involves coagulation lupus tests. You can also have x-rays or scans of the heart, kidneys or other organs. There's no cure for lupus, however symptoms can be managed with anti-inflammatory medicines, steroid tablets or injections, or creams for the skin problems. Avoid sun, pace yourself to avoid tiredness, stay active, and a balanced diet and relaxation. So now we'll talk about acquired immunity in more depth. So we have active immunity. You can have natural active immunity, which develops from exposure to pathogen causing a specific disease, or artificially induced active immunity, which is when an individual is intentionally exposed to foreign antigens for a vaccine to generate a protective immune response. Passive immunity can either be natural or artificially induced. So in natural, antibodies are transferred by natural means from one person to another. This can be from breastfeeding or through the umbilical cord. Or artificially induced is giving someone a transfusion of antibodies produced by someone else. This only lasts a few weeks as there's no involvement of their own immune system. So an immune response to vaccination. Vaccinations are a medical procedure involving the administration of a weakened or dead pathogen, whole or fragmented, in order to trigger the production of antibodies. So we want to vaccinate for immunological memory. This is the ability for the immune system to respond more rapidly and effectively to a pathogen than its previous encounter. So a primary response in its first encounter with a pathogen. First, the pathogen is recognized for the first time. 2, the antigen is identified by B cells and plasma cell is produced for the appropriate antibody. 3, memory cells are produced. And then in secondary response, the next time the pathogen is encountered, the same pathogen is recognised and then higher quantities of antibodies are produced quicker and for longer. Normally this fights off the infection before symptoms are seen. Herd immunity occurs when a significant amount of the population has been vaccinated and is therefore immune, and this makes it more difficult for the pathogen to spread. It's important to protect those who cannot be vaccinated, such as small children or those with weak immune systems. The threshold percentage of the population that needs to be vaccinated is different for each disease. This is calculated using the basic reproduction number, notated as R0. This is how many people a single person could infect. Some diseases are infectious and not contagious, so it does not protect against all vaccinated diseases. Some other factors taken into consideration when calculating this threshold is how effective is the vaccine, how long does the immunity from the infection or vaccine last, and what population form critical links in the disease transmission. Herd immunity. There are some problems faced when trying to get herd immunity, such as some individuals may not produce an immune response due to a compromised immune system. Some individuals get infected before the vaccine has had time to work. The pathogen that's being vaccinated against may go undetected by the immune system. There could be antigen variability, which is when there's different variants of the pathogen all presenting different antigens mutating quickly. There may be individuals who don't want the vaccine for many reasons. There are many vaccination programmes that are available. So the NHS is responsible for England's vaccination programme. Children are vaccinated at set stages throughout their childhoods, either at the doctor's or at school by a health visitor, nurse or GP. Adults aged 50 plus, immunocompromised or disabled gets vaccinated against the flu yearly. Here is some of the vaccination programme for the NHS, what age and what vaccination you would get. You can also get additional vaccines, such as when you work for the NHS you get Hep B. So some factors affecting vaccine uptakes are seasons. What season it is affects the spread of many diseases. Vaccines cannot be given to someone recovering from another illness, and anti-vaxxers believe vaccines cause autism, cancer or other diseases. However, the autism one has been disproven by many studies. There are many benefits of vaccination, such as saving in healthcare, reduction of antibody resistance, extended life expectancy, cancer prevention, economic growth and empowerment of women. These way outweigh the complications of diseases and negative health benefits. And smallpox has been eradicated due to a successful vaccine programme. Thank you for listening to my presentation. I hope you enjoyed it.